Welcome to Beyond J, taking conversations from campus to the world. I'm your host, Philip Allstott. What can the common fruit fly teach us about autism? That's a question that Sacramento State biology professor Dr. Kim Mulligan and her students are working to answer. Mulligan's research centers on the molecular basis of brain disorders. In her lab on campus, she and her students use the common fruit fly to study how genes and environmental factors can converge to cause neurodevelopment disorders, such as autism and Fragile X syndrome. In today's episode, she talks with us about the broader implications of her research and why she's so devoted to teaching and mentoring, as well as a surprising connection between fruit flies and us as humans. Dr. Mulligan, welcome to the show.
Thank you so much. It's really a pleasure to be here.
So, why don't you just tell us a little bit about the research that you do on campus?
I'd love to. So my research here on campus focuses on neurodevelopment. And so I'm particularly interested in the causes of neurodevelopmental disorders. So not just neurodevelopment, but when it goes awry and causes changes in the brain that can affect people detrimentally. And so first, I want to say that, the neurodevelopmental disorders I study cover things like autism, intellectual disability, epilepsy. We study genes that are associated with these disorders. But, these disorders range from very severe to very mild. And so, we've had individuals with autism in my lab who are not the type of individuals who would need treatment or are not interested in a cure.
High functioning.
Those are not the forms of neurodevelopmental impairment that we study.
Okay.
We study forms that cause very severe impairment that significantly affect people's ability to live life on their own independently. And so I use fruit flies as a model organism to study neurodevelopment. And some of the causes that I'm interested in studying are genes, not just genes, how they converge with environmental factors. And part of the reason I think it's important to study environmental factors is because there are meaningful measures we can take to impact people's exposures to environmental factors. So some of the environmental factors that we've studied in my lab are chemicals used in the synthesis of plastics, flame retardants, and right now we're starting a big project looking at air pollutants.
Now, you mentioned that you are using the fruit flies.
Yeah.
And this is wild to me. Like, why are you using fruit flies to study human neurodevelopmental disorders?
So you mean like besides the obvious that they're adorable? So
oh, that's a good reason. No, starting with not
super cute. So fruit flies, actually they've been around as a model organism for over 100 years. So they're very well studied, and that gives us an advantage because there's this vast experimental toolkit that's already been developed, and we know a lot about their genome and how conserved their genome is with ours. So as someone who's interested in studying diseases that affect humans, one thing that's often shocking for folks to learn is that 75% of genes associated with human diseases are actually conserved in fruit flies.
That's wild.
And so, there are a lot of genes that are implicated in human health and disease that the first place scientists started to understand how they function at the molecular and cellular level actually came from studies in fruit flies. So even though in humans, the processes obviously are more complex, right? Our brain is a lot more complex than a fruit fly brain. Let me give you one tangible example. One of the genes we study is called FMR1. It causes Fragile X syndrome in humans, an intellectual disability. One of the phenotypes it causes in neurons in a human is it causes this overbranching phenotype of a structure called dendrites, and they end up making too many connections with other neurons. And this kind of muddies up the neural circuitry and communication. It's kind of like if you ask for directions to a restaurant, it's nice to get 1, 2, 3 options. If I gave you 100 options, that would be confusing, right? And so the neural circuits, you want them to be pretty refined. So if FMR1, this gene, is disrupted, they become unrefined. That same thing happens in a fruit fly. You get too many synaptic connections. So even though it's more complex than a human. That fundamental process that it participates in is the same. And so we can start to understand its role by examining it in a fruit fly.
Okay, so now that starts to bridge some connections for me too, because when I first heard about this, I was like, oh my God, they're like little human brains. Do they have feelings? Are they thinking like we think? But it's not that it's the same brain.
Yeah.
It just works in a similar manner, and you're able to somehow make the connections
extrapolate information.
Extrapolate, thank you.
Yeah, it's, one analogy would be, so right, humans have about 20,000 genes. Fruit flies have, I think something like 13,700 genes, right? So they have fewer genes and they're less complex. It's kind of like if you were to buy like a Crayola marker set, right? It comes in, you can buy marker sets that maybe have 24 markers. And so you have a lot of colors, all the colors you need. But if you want to get fancy, you could buy a marker set with 48, which tends to be, instead of just 2 reds, you have 5 reds, and they're subtly different shades.
Yeah.
So it's kind of how the genome complexity grows from lower to higher organisms. In a fruit fly, you have all the colors there, but they're maybe not as complex, and there's not as many of them.
The fruit flies, though, they're so tiny. I can't imagine how you would get to its brain even.
Thank you, microscopes, first of all. Really useful invention. Yeah. We do dissect the fruit fly brain, both the fruit fly and the fruit fly larvae brain in my lab. So I train students to do this all the time. So you have to get specialized forceps that have very fine point tips, right, and are really sharp. And you have a dissecting microscope. And you have to have steady patient hands.
Oh, yeah.
So if you have had, it's like you have to finely titrate the amount of caffeine you have. You need enough to be alert, but not too much that you shake. Because really, if you're trembling, you're not going to be able to successfully dissect those brains. Yeah.
Yeah. Now, why not something larger? Like, I guess being a lay person, I just think, oh, they're probably using mice.
Yeah, of course. That's such a good question. So when I was an undergraduate, fruit flies were the first model organism that I used. And they are such a nice entry-level model organism, and not just entry-level. I know PhD students, postdocs who work with fruit flies as well, but they're particularly amenable to new students because they're very inexpensive. So it doesn't matter if you mess up the brain dissection 100 times. We have plenty more fruit flies for you to work with. I also did work with mice as a postdoc, and it was so painful for me because they're cute and they're fuzzy, and I really, I was studying brain development. And when you're studying brain development, that means you're dissecting fetal brains. So as I'm pregnant with my son, I'm having to dissect, fetal brains from pregnant moms, and it just didn't.
Well, this thing that can look at you and it's got flesh, right? Like unlike the bug, it's got the exoskeleton.
And even though fruit flies are adorable, this is going to sound awful, I don't mind decapitating them. But and I am very grateful for people who do work with vertebrate model organisms. It's so important to scientific research. It just wasn't for me. It's also really expensive. They bite you. So they're cute and they bite.
Considered that.
I've been bitten by so many mice. Yeah.
Oh no. So let's going back to the research then. Like, what are the potential implications of the research, and just like let us know why does it matter that you're researching this?
Yeah, thank you so much for asking that question. Again, when I was a postdoctoral fellow, I was studying the genetic underpinnings of neurodevelopmental and neuropsychiatric disorders, so things like autism, schizophrenia, and bipolar disorder. But, there's nothing, if an individual has these genes, they have those genes. But we also know that there are environmental risk factors that can increase the severity, or increase risk of neural impairment. And the thing about environmental risk factors is those are things we can control. So by adding to the body of research that says, hey, this, whether it's a particular air pollutant or a pesticide, if these confer risk of neurodevelopmental impairment, we can hopefully leverage what we learn by communicating with policymakers and politicians to say, hey, look at this growing body of evidence that indicates that these, air pollutants are causing great harm to the people in our community. So what measures can we take? Can we put certain emissions filters on cars, or, help install filtration into home filtration systems, right? That will help minimize that exposure risk.
Yeah. I mean, this really feels like one of the major differences when you look back a generation, 2 generations, 3 generations, right? DDT is fine to use, in the early to mid 1900s. And so this is just a continuation of how do we, treat ourselves better, treat the planet better?
Yeah, it's really complicated.
I'm sure.
Yeah, and different governments have different approach to mitigating risk. And so some governments are they are quicker to ban certain chemicals than other governments. And I don't know enough about how lobbying works and, how money incentivizes the choices to allow some chemicals that have been proven time and again to be harmful to humans to still be legal for use in certain industries. But I think that's where the more research, the better, and the more people putting out their findings to the public. In fact, DDT, it's interesting you bring that up. What actually led to the change was a book that was published for that people who were not scientists were reading, and it caused such great alarm that it motivated change. This is the same thing with bisphenol A. The reason we have a lot of BPA-free projects is because there was public outcry when people started to learn of it as an endocrine disrupting chemical.
Or very recently, throw away your black spatula, right?
Yeah.
This has been a whole thing in the news.
Mm-hmm. The thing that's alarming, though, is it's still not, there's no legal repercussions. So, I read an analysis recently of how much BPA was found in athletic clothing. So it's still there, and it's everywhere. And one of my largest studies over the last 7 years was a comparative analysis of bisphenol A with the chemical analogs that are used to replace BPA often in BPA-free plastics. So they're chemicals that look structurally quite similar to BPA, but are flying under the radar of public awareness, essentially. And so I think scientists also, it's upon us to be better public communicators about what we're finding to try to motivate people to change practices, but it's hard. Yeah.
Okay. So let me change gears a little bit.
Sure.
Because I'm always really fascinated to know why people choose to be here at Sac State. Like what's your story? How did you get to Sac State, and why are you here?
Yeah. Oh, yes. So I'm from Sacramento. So I was born and raised in Sacramento.
Great.
I actually started my college journey at American River College, and then I transferred to UC San Diego, where I majored in biochemistry and cell biology, and then spent 2 years as a scientist at the Center for Molecular Genetics at UCSD. I then moved north to the Bay Area. I went to Stanford University, where I got my PhD, and then did a postdoc there, and then at UCSF. And so I went, I had this trajectory of starting at community college and then moved up to, some of the top institutions in the world. And I really wanted to take everything that I learned and bring it back to my home community of Sacramento and serve my community here. I also just love the mission of the CSU. So, there are a lot of universities that will only take students who have, 4.8 GPA. I reject that because I have had so many students who they're just as brilliant as those 4.8 GPA students from high school, but they had a number of barriers in their way.
Yeah.
And I appreciate that the CSU's mission is to educate as many members of our community as we can, right, to elevate our community. And, so, I forget the statistics. It's something like 1 in 20 Sacramentan is a Sac State graduate or went to Sac State.
Yeah.
And so I think it's this really incredible opportunity that I have to give back to my community and to work with a population of students that I feel like I can connect with, right? 50% of our students started at community college. That's my story too. I was a late bloomer academically.
Me too.
I grew up skateboarding and I looked like someone who wasn't going to become a scientist. And so I got a lot of really negative negative feedback when I shared that I wanted to be a scientist.
Yeah.
And it was a few professors at American River College who really believed in me, and I think were the first time I had someone in a position like that see me as someone with potential. And I know what that did for me to help me feel like I belonged in the STEM community. And so to be a person who, coming full circle, now here I am in this role of a biology professor who has the opportunity to help lift up my students. Sorry, I'm getting teary-eyed.
No, it's okay. It's
I
my
can't
Sac
imagine
State
anything.
story too. It's I came here as an undergrad, I think out of convenience, being from Sacramento myself, and stayed for a graduate degree because of faculty that saw something in me and made me aware of that and wanted to cultivate something in me. And that's not what I expected when I came here. Yeah. When I got it, I was like, I need more of this before I go.
It's really an amazing community of educators that we have here. It just feels really special.
It does. It does. So you mentioned working with the students, and I kind of want to dive into this a little bit. So tell me how you involve the students in your research.
My students do everything. So I really want to equip my students with the most competitive skill set possible to help them compete in whatever comes next for them, whether that's a PhD program or a field in allied health or med school, or maybe they want to go straight into industry, whatever. Maybe they want to move away from science. But I believe that doing research can give students so many different skill sets that can help them compete in whatever it is they do, whether it's communication skills, both oral and written, troubleshooting skills, critical thinking skills. So in order to achieve that laundry list of skill sets, I need them to be involved in all aspects of the research. And so, my role in running the lab, of course, I write the grants that funds the research. And then when I recruit students, I actually have more senior students train the incoming students, right? So they get that mentorship opportunity and learn how to mentor and communicate and help new students manage their time. And then students work together in teams on different projects. And so they'll work together on a specific experimental paradigm. They'll meet with me to share how they're doing. I'll help with troubleshooting, but they're also engaging in that troubleshooting, that critical thinking piece to figure out where the kinks are in the experiment. Where did it break down? How can we change up the method? Then I also have them present their findings. So I just had 2 students present their research at the Sac State Research Symposium. I have 4 more going to a conference in April to present their research again. And so they're actually involved, and they present the research much more than I do. So, from start to finish, my students are involved in all of it.
Yeah. That's amazing. I understand. Well, I know that you yourself went to some world-class institutions, and I understand that some of your students go on to go to some pretty impressive places themselves.
Fancy places. That's right. Yeah, I have. One of my former students who I was just talking with this morning, she's a PhD student at UC San Diego. I have students in PhD programs at UC Berkeley, Johns Hopkins. I've had them go all over. And I should say that the conference that I alluded to that they're going to next month is let me get this right, it's WebCirc, West Coast Biological Sciences Undergraduate Research Conference. crushed it.
Yeah, you did.
So when my students go there, they actually compete against students from the big research institutions in California. So including, right, Stanford, Berkeley, USC, UCLA. My students, almost every year we've gone, have taken home first prize wins up against those students from these other
congratulations
institutions.
to you and to them. That's fantastic.
Yeah. I mean, again, I think when you have high expectations of your students and you give them the support they need to achieve those expectations, beautiful things happen. Yeah.
Yeah. Okay. Well, let's switch to some lightning round questions.
Oh no. Okay.
Yeah. We'll make this fun before before I lose you for the day. I also understand that you love to travel and that you'll be going to Kenya soon.
Yes, I'm so excited.
I'm a little bit jealous about that, but why don't you tell me, like, what is your favorite place that you've ever been to in your travels?
Oh my gosh, this lightning round's going to be terrible. I can already tell my kids. It's funny, my kids ask me for favorites all the time and even favorite color. I'm like, I don't know, the pressure.
Okay, how about this? Where are you eager to return? Because I know, like, I lived in China for a while. And I have dreams about it, and then I wake up trying to, like, scheme, like, how can we get back to China, even just for, like, a vacation?
I know. We just did a trip to Thailand, and that was I'd gone to Thailand with my husband 19 years ago. And so, you know, this time we took our kids, and it's just such a magical, beautiful place. I could eat Thai food for the rest of my life, from, you know, the coast, the islands, to the hill country. I mean, it was just beautiful. The temples, I could go on and on. I feel like I want to pull out my phone and do a slideshow for you right now.
When we turn off the microphones, you could show me some pics.
But I'd say also, like, just nearby, a place we go back to all the time is Point Reyes National Seashore. It's just such a treasure. I love that place.
Yeah, me too. Okay, and you're a Kings fan, yes?
Huge Kings fan.
Okay, well, who's your favorite player?
Oh my gosh, with the hard
I know, with the favorites,
questions
right? I know you
again.
warned me, but that's what I had prepared.
Okay, I would have to say right now, DeMar DeRozan, Keegan Murray, Keon Ellis, and DeMontis Sabonis.
Okay.
Can you?
You really narrowed it down.
Sorry.
But what if I opened it up to, like, all-time favorite Kings player?
I'd have to go with Bobby Jackson. Big game Bobby.
Okay, good.
Love that guy.
Yeah, me too.
My son actually had the opportunity to get coached by him, which I was just such a fan. We walked in, he had a training camp for kids, and walked in, I saw him, I was like, my son's like, hey, Coach Bobby. It's like, oh my gosh, my son's
He's
on
such
first
a
name.
nice guy,
He's
right? I mean,
so nice. So nice. Yeah.
Okay, well, it's been a blast, and I really appreciate you coming down here to talk to us.
Oh, so much fun. Yeah, thank you.
Thanks. Beyond J is a production of University Communications at Sacramento State. Our producer is Jonathan Morales. Photography and videography by Bibiana Ortiz. Additional research by Cynthia Hubert. And editing by me, your host, Philip Allstott. If you liked this episode, please leave us a review and consider following us on your favorite podcast platform. If you have questions or comments, email us at communications@csus.edu and visit us online at csus.edu/news/beyondj.